All right, I think I'm live. Somebody would give me a comment just to kind of let me know if this is actually working or not. Anyways, so all right, so I think I got a second or two lag, but it shouldn't be too terrible. Anyways, so last time that I made a video, I was talking about building a Trinity assembly. So we're using Trinity to make this work. And let's look at some of that output. So this is going to be a test mouse, data, seeks. All right, so I have my output directory. So I have my Trinity output that's going to have all of my Trinity stuff. I know I kind of already walked through this already. Most of this is garbage that we don't need. A lot of this stuff we could get rid of. The only thing that we care about is this Trinity FASTA is this Trinity FASTA file. And so this is going to have all of our transcripts in it that we care about. Right, so it has a bunch of different names, Trinity, some name, how long it is, and the sequence itself. Overall, let's see, so I have, oh, that didn't work. Let's see. All right, so I have 132,000 sequences. Sorry, I think I'm having some connectivity issues. Hopefully this won't be too much of a problem. If it is, I'll find a fix. But hopefully this is working again. So let's see. So again, the Trinity stuff, the Trinity output is unannotated. It's just our transcripts. We don't know what genes are what. We don't know where the start codons are. We don't know where the stop codons are. We don't know what's an actual protein coding gene. We don't know what's a long non-coding RNA. We don't know what any of this stuff is. And so the next step to deal with an assembly is to actually annotate it. Tell us what genes there are, where they are, and how many different isoforms are present, how many transcript variants are present, things of those sorts. Those are the kinds of questions that we need to try to answer with our Trinity output. So the best way to do that there are a handful of tools already developed by the Trinity team to help annotate their product. So one of those tools that I'm keeping in opt is called transdecoder. So what transdecoder does is that it's going to look for open reading frames in our assembly. So it's going to look through every single transcript and decide, hey, is there an open reading frame? If not, if so, is it complete? Is it just kind of like the 3' end? Is it the 5' end? Is it just an internal sequence, etc.? So it's just going to see what's actually present. And transdecoder really isn't that hard to run. Let's see. So all that would be done is to call the program. So opt transdecoder, transdecoder long ORFs. So long ORFs just stands for is going to look for long open reading frame. And even the help manual is already pretty polite as soon as it's opened. All of that, the only thing that we have to provide to this program is this -t and then providing a list of transcripts or the sequences that contain all of our transcripts. In this case, it would be the Trinity.FASTA. a protein or a protein coding gene if 100 amino acids are actually present. If not, it's going to be thrown away. Probably the other important one to look for is the genetic code. So more often than not, for like everything, like 99.9% of all genes, they're all universal. So ATG to stop, they have this all universal um, protein coding translation table. But if we're dealing with mitochondria or some of the rarer things, they will have a slightly different translation table. So if that's the case, we would need to submit that. Like what organism, what translation table do we need to work with? And so these are kind of like the other translation tables, but by default we use the universal. So again, if we're just doing this, we would just run this long orfs -t on the trinity.fast output. So that's going to, what that will do is it will give us this directory, trinity.fast transdecoder.dir. And so all of this stuff is our, it's going to have annotations in it, at least in terms of um, whatever. It's within this, it's going to store the output of transdecoder. So let's kind of take a look at this as well. So it's going to produce 4 different files. It's going to produce a longest orf.cds, a longest orf.gff3, and a longest orf.pep. The base frequencies we don't need to worry about, that's used by the program. It's not all that super important. What are important, well, probably the most two important files are the longest orf.cds and the longest orf.pep. So let's see. So we can see how many sequences are actually present in these FASTA files because they are FASTA files. CDS. So that is a FASTA file. It just tells us within it, it tells us the transcript name, and it tells us what kind of annotation it is. And in this case, it's an internal, which means it was not able to find a start codon or a stop codon, but it was able to find a string of about 100 amino acids that are uninterrupted. So it probably is a gene. Again, some of these transcriptome assemblies, they're good, but they're not perfect. And a lot of the stuff we do in biology, we can't get it too perfect. But let's see. So what we would find in these headers, let's see. So we'll see internal, we'll see a 5' partial, a 3' partial, and complete. And so what all of these means, again, internal is like saying we found this long string of amino acids, but we couldn't find a start codon, we couldn't find a stop codon. Complete means we found a long string of amino acids plus the start codon and a stop codon. So this is probably a complete gene or close or really close to it. 3' partial means that we were able to find, or what it's saying is that what's not complete is the 3' region of the gene. So we've been able to find a start codon, we find a long string of amino acids, but a stop codon wasn't present. Again, this could be a true biology thing, or this could be like an assembly artifact. It didn't assemble the entire transcript. And so generally what we want to look for are the complete ones. These are probably the most helpful. They're probably real genes that probably have real annotations. But nonetheless, all of these different, those are the 4 main types of annotations that are outputted by transdecoders. So let's look at this for a second. So if we look at let's find a 5'. So here's a 5' partial. Again, what it's saying is that the 3' end of the gene was actually assembled. What it couldn't find was the 5'. So we don't see a start codon. but we do see a stop codon. So TAG is a stop codon. If we look at the 3' partial, it finds the start codon, but what's incomplete is the 3' region, so we don't have a stop codon. Let's see if we can track down on complete. All right, so here's a complete transcript that has the start codon and the stop codon. So this is probably a pretty good gene. Anyways, so again, we may want to count out how many how many transcripts were actually annotated by transdecoder. What genes actually seem to have an open reading frame on it. So let's see, FC on the open caret. And so it's going to tell us 75,000 transcripts had a protein coding gene on it. Remember, we had about 132,000 transcripts assembled, but only about half of them appear to be protein coding. The rest could be non-coding RNAs, or they could be transcript assembly artifacts, just things that were developed by the assembler, but not actually, not really a gene, right? Sometimes assemblies do weird things. All right. So we have our longest CDS, we have our protein coding gene, we have our CDS genes. So the PEP, what's in the PEP is the same stuff as the CDS. It's just going to be the translated amino acids um, given the universal uh, translation table. So again, let's look at the PEP. So here's again, just to reiterate, here's a 3' partial, which means we don't have a stop codon, but we do have a methionine start codon. Yay! In the 3' partial, it's not going to give us a stop codon, but it is saying the stop codon was present. Right, let's see. So it's going to be the exact same sequences. So if I grep C on all of this stuff except for the GFF on the open caret, it should give us, right, it gives us the exact same numbers, the exact same number of sequences that are in both. It's just one's the CDS and the other is the amino acid sequences. The longest orf.pet, or sorry, longest orf .gff, this just tells us locations of these genes. So all is it saying? It's saying on this specific transcript, it found a gene starting at base pair 1 into base pair 417. Right, that's all it is. It's just giving us a location of that annotation. It's not telling us what the gene is. It is telling us a little bit more information, like it is an internal. Right, so the whole transcript looks coding. It's just that it's lacking. It wasn't the start codon, the stop codon, and the UTRs, the 5' and 3' UTRs weren't actually present. Let's see if I actually find it complete. Whatever. All right. So again, we have our transcripts, but we don't know what these genes are. We at least know which ones have coding regions on them, but we don't know what the genes are. We don't have any good annotations. And so in the description here, I do have a link to a page that describes a pipeline to annotate Trinity transcripts. And so it's there's a lot of different ways that we can look for. So we can look for transmembrane domains. So these will be like long chains of amino acids that that will be typically hydrophobic, and which means that this protein is probably anchored into the cell membrane or some membrane. And so transmembrane domain finder will look for those and at least can tell you if a protein is bound to a membrane or not. There's other things we can determine if a protein belongs to a specific gene family using PFAM, but probably the simplest starting point is to use similarity, find genes that are similar to our transcripts using BLAST. That's probably the go-to way to at least start. And so the link in the description is for Trinitate, and Trinitate has a whole lot of different steps. But probably the first step is to actually blast it to the SwissProt database. And so remember, our SwissProt contains a lot of information, a lot of annotation information, like where gene is expressed, um, what tissues it's expressed in, where is it expressed in a cell, and what functions does it seem to have. And so I downloaded the SwissProt database, and so that's going to be stored in the root data. Same wherever let's see, it's the same wherever I put the Bush baby CDSs back a while ago. I just created a directory in SwissProt that you can access and go to. And so what's in here, the main file that's in here is this uniprot sprot.fasta. Let's see, there should be over 300,000 sequences in this by this point. All right, so we're at 500,000 pretty well annotated protein sequences. If you look at this, they're all protein sequences. And so our SwissProt, right, these are just all the proteins that are available in SwissProt. And they do have some amount of annotation information. Not necessarily in this database, but we could use these numbers. So this number, Q number, can be used to find additional annotation informations associated with like gene ontology annotations. Let's see. So it does tell us a lot about what animal these proteins will be found in. So it should find mouse. If not, nope. Whoop. Let's see. Mouse. All right. So there's a mouse gene. Protein beta alpha makes me think that this is it's a globin gene, probably. Mus musculus is a species. So our SwissProt contains a lot of information about what species these genes are from and what do they do. So we want to compare our transcripts, not only just our transcripts, our open reading frames, to the SwissProt database to see which proteins are actually present in our transcriptome assembly. So I'm just going to go back to the transcriptome file. So where the output of transdecoder is at. So that's mouse data seq liver trans. And so whenever I want to run blast, this one will take a while. So running blast on 130,000 sequences will take a considerable amount of time. So I'm not going to make it run for the whole time, but I'm going to let this output like 100 annotations. And the actual line that you want to be worried about is to run blast p. So we're going to run blast p, and a blast p is having it will take a protein query and look through a protein database. And for it is a little bit slow, but it is probably the way to start. So we'll blast p again. So the NCBI, I love the NCBI, and I also hate the NCBI. And one reason I hate the NCBI is because they do not really annotate their help menus. So, right, so their help menus aren't exactly very user friendly. This is the main blast help page. It tells us absolutely nothing except the parameters that are available, but it does not tell us how to use them. Kind of frustrating. But I'm going to save you a lot of time. So again, we're going to do a blast P. So there are a lot of different blasts. So we can do a blast in, which is nucleotide to nucleotide. We can do a blast P, which is protein to protein, or we can do a blast X, which is nucleotide query to a protein database and variations thereof. But since we're doing a blast P, we're going to do protein to protein. So our first option is the query. And so the query is going to be our transcript file. So longest.orf.tep, right? So this has to be the proteins. So we're going to do a protein blast. So it has to be the amino acid sequence. And the database that we're looking for is what we just looked at, the SwissProt database. So that's data SwissProt Unipod.fast. And remember, there were several different files in that directory. Um, there's only one that matters, and that's the Uniprot SwissProt.fasta. Any all the other files that are in that directory are associated with running Blast or Blast reading those files. Whoops. So that's our DB. We need this out format 6. What that means, out format describes how we want our output to look like. And out format an out format 6 just means that we want our output in a tab delimited format. Max target seeks 1. So what max target seeks is going to do is for every transcript, it's only going to give us the first blast hit. There's definitely a lot of discussion among bioinformaticians, and usually you don't want to use a max target seeks 1 because what it's going to do is give us the first hit. And so the first hit may not always be the best hit. So sometimes it would be better. This is a nice shortcut. It's a parsimonious shortcut that works, 90% of the time. But if we were trying to get this as the best annotations we could, we would let this annotate as many regions as we can, and then go through it later and pick the best one. But that takes a little bit of coding. We're not going to worry about it. We're just going to run a max target seeks 1. Num thread. So this is a how fast we want this computer to run, or how many processors we want to let Blast use. For my case, I'm going to pick 10, and an e value. So we want an e value threshold. So 1 e to the negative 5 is pretty accurate. So what this means is, two sequences can be similar by chance. And so what we're doing is applying a p-value, the equivalent of a p-value. And so if we have an annotation that gives us an e-value of 0.001, well, that doesn't pass the actual threshold to report. All we're going to report are the hits that have this e-value that's less than 1 times 10 to the negative 5th power. So fairly unlikely sequences to be similar by state, not necessarily similar through homology. I think that's all the parameters I need, so yeah, that's fine. And again, this will take a long time to run for 137,000 sequences, so I'm just going to pull out the first 100 just to actually look at it. So this will take a couple of seconds. Yeah. Okay. All right, so these are just hits for the first 100 sequences, the first 100 transcripts in this. So this output is tab delimited, so we do have these columns. It is a column. And so our first column is the transcript name. So this is from our Trinity/transdecoder outputs, and over here is the hit. So this is what was found in the SwissProt database. So we have the query, we have the hit, we have a percent similarity. So this is 100%. These two sequences are 100% identical to one another. That length of similarity is for 138 amino acids. There were 0 gaps, 0 mismatches, and these two values. So this is the start of the hit or start of the query. So query base 0 to query base or to query amino acid 138, and this is the region that was hit in the SwissProt database. So this is the the database hit where this sequence was similar in the database. So this sequence from amino acid 451 to 588. And then there are e-value. So e-value of 3.8 times 10 to the negative 89. Pretty low, right? They're right. This is what you would expect of an e-value or of two sequences that are 100% identical is a very, very low or zero e-value. And this last number is a bit score. And what bit score does is it is this value of similarity. And its best use is if there's a tie. So if you have two sequences or you have a sequence that hits two genes, and the e-value is identical, say it's 0, but the hit with a higher bit score is the hit that you would choose, but we're not necessarily dealing with this. As you see, as we would expect, is that all of our transcripts generally are hitting on mouse genes. We would expect we have mouse assembly, we would expect mouse genes in it. And most of them, not all of them, but most of them are 100% or close to it in similarity. That's something that we would expect. Again, these are mouse genes. We're hitting to a mouse database. Mouse is a very well annotated source of information, so you would expect this to be all pretty similar to one another. That doesn't mean that's always the case. Sometimes we do get some pretty terrible hits, right? So that's a 26% identity. That may or may not be true. There are ZNs all over the place. These are like zinc finger proteins. They're DNA binding, really common in the genome. Sometimes they're a lot of them are uncharacterized, right? We at least know they have these specific DNA binding domains, but we don't know what they do. But yeah. And you will see sometimes on occasion. So we have two different transcripts that are hitting on the same gene, right? So here's one transcript variant hitting on that gene, and here's another transcript variant that hits the same gene. That's not all that uncommon. Again, sometimes we see some weirdness in assemblies, but whatever. I think that's it for now. On Wednesday, I'll talk more about how we can use this blast information, like how many sequences are actually annotated, how many different annotations we see, etc. So it's not necessarily using a tool, but using our tools that we've already learned, grep, sort. We haven't learned sort, but grep and cut. We'll deal with it. All right, I'm going down now.